Hey everybody, welcome to Chem 103 again. Today we're going to be talking about our biodiesel synthesis that we did last week. So last week we kind of just did the lab and then that was it, and I just said, follow the directions and we'll talk about this later. Well, welcome to the later. Today we're going to be looking at the actual chemistry of what's going on in that reaction, and that will inform what we do and how we understand this process going forward. So before we can get right into the reaction from last week, I have to tell you a little bit about some of these molecules involved that we haven't talked about yet. So just a couple things to review quickly. Remember that I am, as we talked about, I guess it was 2 weeks ago, for the remainder of the semester, when we talk about these types of organic molecules, we're going to be mostly using these line or skeletal structures, so make sure you go back and review that if that was something that you struggled with in the last module. So before we get right into the biodiesel, I want to talk about a couple types of molecules that we're going to see and how they interact with each other. So the first one is fatty acids. And fatty acids are biomolecules that have some particular properties. So I'm going to draw one here, and then we'll talk about them. All right, so here's an example of a fatty acid. And a couple of things you should notice here. One is the long chain, and if you remember when we talked about line structures, that long chain is actually carbon atoms all connected. So just to kind of refresh, remember that when we have this kind of zigzag shape, that what we're really talking about is CH2, 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 and so on. And then this one at the very end would be a CH3 because we talked about how carbon needs to have 4 bonds, so we assume those extra hydrogens. Again, go back and review if you haven't seen that one yet, or if you haven't mastered that stuff yet. And then over on the left, we see this, this functionality here. We haven't talked about functional groups, but this is called a carboxylic acid. And that's where the acid part of fatty acid comes from. And that's what's going to allow it to connect in the ways that we're going to talk about. So fatty acids are a member of the macromolecules class of lipids. We'll talk about that a little bit more when we get into macronutrients next week? 2 weeks. And they're all going to have the similar structure with this acid on one side and then the chain of carbon atoms. But there's a couple different ways they can have this structure. So the one that I have drawn here would be an example of a saturated fatty acid. And we can contrast that with what we would call an unsaturated fatty acid, which is where we have double bonds within that carbon chain. And what those double bonds are going to do is not only add the double bond, but they're going to change the geometry by making that fatty acid sort of turn a corner like this. So this would be an unsaturated fatty acid. I made some models here so we can kind of see what that looks like. So here is, let's see, do I have this? I have it flipped relative to that. So here's the example of kind of what I drew. I didn't make the chain quite as long. Fatty acids generally have anywhere from 12 to 20 carbons, but I didn't want to put those many pieces together. So we have something like this, but you can see the H, O, and then the double bond to oxygen, and so on. So you can see that this chain has kind of free rotation to flop around as it will, and if it were much longer, it could do even more of that. And we draw it in this stretched out way. But then if I add a double bond in the middle here, and the way this particular model kit works, you add a double bond by taking out one of these and putting in these blue ones. Since you're not here to try this, if you would like to try it during lab, when we have some downtime, just let me know. I'll get one of these kits out for you so you can play with it a bit. But what you find when you play with this is there's two ways you can put this thing on here. You can do it like this, which causes it to turn a corner, and you might say that the two carbon parts are on the same side. There would be two hydrogen atoms, coming out this way. And that's kind of what I've drawn, that it makes the chain sort of kink around. That orientation of a double bond is known as cis, for the same side. But we could also arrange this the other way, so that it continues that zigzag pattern, like that. And if we were to draw that over here, so we'd have basically the same thing, but now the double bond is like this, and that's known as a trans double bond, because now you can see those hydrogens would be on opposite sides, like here and here. Whoops, you can't see what I just wrote. Sorry, let me put that back. Okay, now. So you can see that now, the description of what this structure might be like. So unsaturated fatty acids, cis or trans, the majority of naturally occurring unsaturated fatty acids are cis. Trans fatty acids generally come from hydrogenation processes, which used to be used in foods. They're not so much anymore because we know they're real bad for you and not really necessary. Let me rebuild my model here. All right, so that's the basics of fatty acids. Now, fatty acids are not what we call generally fats. Fats come from when these fatty acids connect with a molecule of glycerol or glycerin. So let's look at that one next. Glycerol is a molecule. I'll draw it first in a condensed structure like this. And in a line structure, various ways we could draw that, but maybe something like this. Right, so we have our 3 carbons with an oxygen, with an OH on each carbon. I have a model of that here as well. You can see that one. And so fatty acids can combine and connect to glycerol to make glycerides. And you can have monoglycerides, diglycerides, triglycerides based on how many fatty acids connect to this molecule. And what we think of as a fat, which is actually our starting material for the biodiesel synthesis last week, is a triglyceride. So a triglyceride is taking glycerol, or glycerin, same thing. Actually, I'm going to draw it this way. And connecting to these fatty acids, and I'm just going to draw saturated ones, but it could be whatever. Sorry for my scribbles here as I run out of room. But hopefully you can see that you've got the glycerol molecule here in the middle, and then you have these fatty acids coming off going to either side. So I'm going to build that with my models here too. I've got 3 fatty acids and I've got glycerol, and so I'm going to take, so you'll notice if I want to connect the fatty acid, okay, we'll just use the middle one because I've got it kind of lined up. Nope, got it backwards again, sorry. We got it lined up now. So if I want to connect these to make the structure that I've just drawn, you'll notice there's actually too many atoms here. So I'm going to remove 2 hydrogens and an oxygen, which you might also recognize as water, right? 2 hydrogens and an oxygen. Get rid of that, and now I can connect these here. All right, so now I've made a monoglyceride. I've connected 1 fatty acid to my glycerol molecule here. And let's actually draw that reaction. Again, we're not quite to what we were doing last week yet. This is sort of the opposite of that. It's the connection reaction. I'll draw this glycerol molecule like this, and I'll draw that fatty acid molecule right next to it. I know I'm not being consistent necessarily with my numbers of carbon, but we'll just assume it's some carbon chain. And then in order to get those to connect, I have to get rid of water, which is what I did in my model. So that's here, 2 H's and an O, that's H2O, that's water. So if I get rid of water, I can now connect this oxygen to this carbon. Everything has the right number of bonds. And I've made this connection. All right. Now that's called a condensation reaction, or sometimes it's called a dehydration reaction, showing that water is coming out of there. Right? So water here is a byproduct of this reaction. Now this doesn't just sort of happen like this. You can't just mix up some glycerol in your fatty acid and expect this condensation to occur. Experimentally it takes a little bit more, which we saw last week. We did something a little bit different, but this is the basic idea of how those molecules connect. So if we do that 2 more times, we end up with this triglyceride. So I can do that here. Same thing again, I'm going to remove 2 H's and an O, make my connection, and then one more. Okay, and here we go. It's pretty, even with just a few carbon atoms that I built here, we've got a pretty big floppy structure, but we can see the glycerol right in the middle here, coming out and connecting to these other molecules. There's a nice activity that we might do in lab if we have time, where you'll have a chance to build these things kind of at full scale and working together in relatively large groups. But as you can see, it starts to get a little bit unwieldy as you get bigger and bigger with these models. Okay, so we finally made it to the starting point of our reaction last week, because of course we started with a fat. We started with vegetable oil, and we reacted it to form this mysterious substance that we haven't really talked about structurally yet that we called biodiesel. So let's look at what's going on there. Let's see if I can do this. Can I select this whole molecule? I can move it. I don't know how I copy it. I haven't tried that before. Maybe I can't do that. All right, I'll just redraw it. Okay, so we've got our triglyceride that we're going to start with, and then we're going to do this reaction and make biodiesel out of it. Okay, so here's our initial biodiesel molecule. Or I'm sorry, not our initial biodiesel molecule, our initial fat molecule, our vegetable oil. And actually, let's be a little bit more specific now. The vegetable oil is primarily made out of these unsaturated fatty acids. That's one of the reasons that it is generally a liquid at room temperature, has weaker intermolecular forces. We're going to talk about that next week. So let's actually draw those unsaturated fatty acids coming off of here. Okay, so something like that. There's our vegetable oil. I'm going to put quotes on that because, of course, vegetable oil is a natural product. It's got all kinds of different stuff in it. It comes from a plant. It's a mixture of many different types of triglycerides, but here's one that we're going to say is representative of the whole thing. So that's our vegetable oil. All right, so in the course of the synthesis, what we did was we reacted this with two chemicals. Hopefully you remember they were the ones that we talked about have certain hazards that we had to watch out for. They were methanol, which has a chemical formula of CH3OH, and sodium hydroxide, NaOH. And that's it. That's all that was really added into that reaction mixture. Then you remember you heated it up to 55 degrees C and reacted for 1 hour. So that was the reaction. We took vegetable oil, we added methanol and sodium hydroxide, reacted at 55 degrees C for 1 hour. And so what did we get? So I'll tell you what we got, and then we'll go back and see if you can figure out what had to happen chemically for that to be a product. So we got a couple different products. One product we got was glycerol. Sorry, I'm being inconsistent with the structure here. All right, so that's one byproduct here. We also got biodiesel, which we can show like this. Again, it's going to be a mixture of different compounds, but they're all going to have the same basic structure. And then another name for biodiesel is FAME, which stands for fatty acid methyl ester. So FAME. All right, so that's the product here. And that's what we're going to be analyzing. That's what we're keeping after doing the wash this week. And there's some other minor byproducts here as well, but let's just focus on these as the main ones. So let's go back and look, see if I can zoom this out a little bit so we can see the whole process. All right, so can you start to see how the vegetable oil became this molecule down here? So hopefully you can see that this fatty acid is the same basic structure as we have down here, but instead of being connected to the glycerol, it's now connected to this methyl group. All right. And so that actually comes from the methanol. So if we use color here to show our reagents as they were above, it's actually the methanol that provides that CH3 and O to do that connection. So it's going from this type of a connection to this type of a connection, which, if you notice, is chemically very similar, right? COC. COC. So what's happening here is known as transesterification. The sodium hydroxide is facilitating this transfer of the connection here off of the glycerol and onto this CH3, which is known as a methyl group, which comes from that methanol. All right. We're not going to go through the exact process of how that works, but basically the sodium hydroxide helps break this up. and then it's stabilized by the methanol. So, not stabilized exactly, but I don't know what the right word is there. So these are essentially our two portions of the product that you saw in lab. Well, half of you saw it yesterday, and the rest of you will see today. When you go in and you'll see that there's a nice layer separation there. The top layer is the biodiesel, the product. The bottom layer is the glycerol. And there's also other stuff in there. We used a large excess of methanol and sodium hydroxide, like more than we need. So there's some of that left in there too. We're going to drain that waste and try to purify the biodiesel for later study. So just a couple things to note on the structure here. Why we do all of this instead of just using the fatty acid. So here's the fatty acid, and then over on the left we have the fatty acid methyl ester. What's the difference there? Well, the difference is that when we have this extra little piece on here, now it's going to behave more like a fuel. It's going to be less viscous. It's going to have a lower cloud point, which is where it starts to crystallize when it gets cold. It's not going to clog your fuel lines. If you tried to put something like this fatty acid in a car and actually run it, chances are everything would kind of get gummed up once we get into winter type temperatures. But once it has this methyl group here, it changes its properties and allows it to do that. And that's kind of a little bit of a tease for what we're going to be talking about next week, which is the physical properties of organic molecules related to their polarity and intermolecular forces, so how these molecules interact with each other. That will also help to explain why we make two layers, the top layer of the biodiesel and the bottom layer of the glycerol, along with some of these other excess waste products. All right, so that was kind of a long thing. What I want you to be able to do is to draw these types of molecules and these types of processes. So if I asked you, if I gave you a couple molecules and asked you, hey, can you draw a condensation reaction between these? You could show how those go together and do that. And there's some practice links on Teams for that also. And then as I'm asking you about your biodiesel synthesis, do you know what types of molecules are involved? Do you know what kinds of connections are made or broken and why? And so if you have questions about that, please let me know. We'll keep talking about this. This is a longer term project, as I told you, so we have a couple more weeks of washing the biodiesel, then we'll do our final analysis and calorimetry, and that's usually kind of fun. So let me know if you're having trouble with this or if you need more instruction. If these things all just look like crazy zigzag lines and you have no idea what's going on, you missed last week when we talked about how to draw all those structures and what that all means. So please go back and review that. All right, well, I will see you either later in lab today or in lab on Monday. Have a great week.